Something was buried in the desert sand of Peru that scientists were never supposed to find. Not a treasure, not a weapon, not a secret message carved into stone. Something far smaller, far more fragile, and infinitely more powerful. A molecule, a strand of biological code so ancient it had been locked inside human bone for centuries, waiting in silence while empires rose and fell above it. And when a team of researchers finally cracked it open, what came out didn't just answer old questions, it demolished them entirely and replaced them with something no one on Earth was prepared for. We think we know how the Americas were populated. We have textbooks. We have maps with bold arrows drawn across the Bering Land Bridge. We have a story that's been told and retold for over a century, refined and polished until it felt bulletproof. The story goes like this. Somewhere between 15 and 20,000 years ago, a group of ancient humans crossed from Siberia into North America on a frozen corridor of exposed land. They moved south, they multiplied, they spread, and eventually their descendants became every indigenous civilization from the Arctic to the tip of South America. Neat, clean, settled science. Except it isn't settled at all, because what scientists just pulled out of ancient Peruvian remains doesn't fit that story. It cracks it. And the deeper researchers dig into the genetic code of Peru's ancient dead, the more the old model starts to look like a map drawn by someone who had never actually visited the territory. Let's start with what was actually found, because the discovery itself is almost cinematic in how unexpected it was. A research team led by archaeologist Jacob Bongers from the University of Sydney spent years excavating burial sites in the Chincha Valley, located along Peru's southern Pacific coast. This valley was home to one of the most powerful pre-Inca civilizations in South America, the Chincha Kingdom, a maritime trading state that controlled the coastline between the 13th and 15th centuries. These were not simple farmers or isolated mountain tribes. The Chincha were sophisticated navigators, traders, and political actors who operated vast networks of commerce across the Pacific coast, long before the Inca Empire absorbed them by force in the 15th century. When Bongers and his team excavated the burial sites, they recovered the remains of 21 individuals. These weren't randomly selected skeletons. They were carefully chosen samples from multiple burial contexts, including what turned out to be something archaeologists had almost never documented before in this region, a family ossuary, a dedicated chamber where related individuals were buried together across multiple generations. That single feature alone was a bombshell. But what came next made the ossuary look like a footnote. The team extracted genome-wide ancient DNA from all 21 individuals and ran full genetic analysis. Published in Nature Communications in 2026, the results were jaw-dropping in the most literal sense. The people buried in the Chincha Valley in southern Peru did not carry the genetic ancestry you would expect from people who lived in southern Peru. Their DNA matched the genetic signatures of populations who lived on Peru's north coast, more than 700 kilometers away. Not nearby cousins, not trading partners who occasionally visited. These people were direct biological descendants of northern coastal communities who had packed up everything they owned and walked the equivalent of the distance from London to Berlin across some of the most difficult terrain on Earth, navigating desert coastlines, river valleys, and geographic barriers that would have made every kilometer an ordeal. 700 kilometers on foot, centuries before the Inca built a single road. And here's the part that rewrites the textbooks. The earliest migrants in that data showed almost completely unadmixed ancestry, which means when they arrived in Chincha, they didn't immediately blend with the local population. They stayed distinct. They kept their bloodlines separate. They maintained their identity as a people for generations while living surrounded by a completely different culture. Think about what that takes. That is not the behavior of scattered nomadic bands drifting aimlessly. That is a community with a deliberate, powerful sense of self, a people who knew exactly who they were and where they came from, and refused to let that identity dissolve, even when surrounded by strangers in a foreign land. Only in later generations did the DNA show signs of mixing, as northern migrants slowly intermarried with neighboring coastal groups from both the central and southern coast. And even then, something remarkable persisted. Certain cultural practices, cranial modification, the ritual application of red pigment to the skulls of the dead, continued in the Chincha Valley for at least 2 centuries after the migration. These weren't random habits. These were identity markers, signals to the world that, even as bloodlines blended, something of the original northern heritage was being deliberately preserved and honored. A 16th century Spanish chronicler named Pedro Cieza de Leon actually recorded that the earliest Chincha people were led by a captain who came from far away. That account was written off for generations as legend, a mythologized origin story with no historical basis. Science just proved him right. 500 years after Cieza de Leon put pen to paper, ancient DNA confirmed that a people did indeed come from afar and settle the Chincha Valley, exactly as the old chronicle described. Now let's zoom out. Because this Chincha discovery is one thread in a much larger, much stranger tapestry that ancient DNA is currently unraveling about the Americas. To understand why this particular finding is so seismic, you need to understand what scientists thought they knew about pre-Columbian South America before the DNA evidence started coming in. For most of the 20th century, archaeologists and anthropologists operated on an assumption that pre-Inca coastal societies were largely isolated from one another. Yes, there was trade. Yes, there were cultural exchanges. But the prevailing model imagined these civilizations as essentially local. People who were born, lived, and died within a relatively small geographic range, shaped by their immediate environment. Long-distance migration was seen as exceptional, rare, driven only by catastrophic circumstances. The idea that ordinary people were voluntarily trekking hundreds of kilometers to settle new territories, maintaining their cultural identity across generations while doing so, simply wasn't part of the accepted framework. The DNA evidence obliterates that framework, and it raises an immediate burning question. Why? Why did people leave their homes on Peru's north coast and walk 700 kilometers south? What were they running from, or running toward? The researchers identified multiple possible answers, and every one of them is fascinating. Climate instability is a leading candidate. The Peruvian coast is at the mercy of El Niño, the periodic climate phenomenon that still today triggers catastrophic floods, droughts, and fishery collapse along the Pacific coast. A severe El Niño event in the 13th century could have made the north coast effectively uninhabitable, forcing entire communities to uproot and search for more stable ground to the south. The expanding Chimu state, a powerful northern polity that at its peak controlled over 1,000 kilometers of Pacific coastline, may also have played a role pushing smaller communities out of their territories through military pressure or political absorption. And then there are resources. The Chincha Valley and its surrounding region held access to seabird guano, one of the most powerful natural fertilizers in the world and enormously valuable in Andean agriculture. Copper mines and cinnabar deposits near Huancavelica were also within reach. People walk incredible distances for economic opportunity, and always have. But here's what's haunting about this story. One of the individuals whose remains contributed to this research was a woman. She was buried with spinning tools wrapped in yarn and cotton, objects that speak to her daily life, her craft, her identity as a maker. Red pigment marked her cheek centuries after death, that northern cultural signature applied by people who still remembered where they came from. An examination of her pelvic bones showed signs that she had given birth, somewhere in that Chincha Valley, during a migration that displaced her entire community. This woman had a child. She lived, she worked, she created, she brought new life into the world, and she carried her identity with her until the very end. Co-lead author Jordan Dalton described looking at those bones and realizing that the very personal and physical experience of giving birth had been written into her skeleton, still readable 800 years later. That is what ancient DNA does that no other science can. It doesn't just reveal populations and statistics. It recovers people. Now let's go even deeper, because the Chincha Valley study is actually the most recent in a series of DNA discoveries from Peru that have been steadily dismantling our assumptions about the ancient Americas over the past decade. And some of what came before is even more disturbing to the established order. In the 1920s, a Peruvian archaeologist named Julio Tello excavated a massive burial site on the Paracas Peninsula along Peru's southern coast. What he found inside more than 300 tombs were human remains, unlike any seen before in South America. Individuals with skulls so profoundly elongated that they seemed barely recognizable as human. The Paracas skulls, as they came to be known, are the largest elongated skulls ever documented anywhere on Earth. Their cranial volume is estimated to be up to 25% larger and 60% heavier than conventional human skulls. For generations, scientists explained this away as the result of cranial binding, the practice of wrapping infant skulls tightly with cloth to reshape the growing bone over time, a practice documented in various ancient cultures worldwide. But when DNA testing was conducted on these skulls, something unexpected emerged. Mitochondrial DNA extracted from samples including hair, teeth, and bone fragments revealed haplogroups, genetic family lines, that don't belong in South America at all. The most common non-native haplogroups found were H, H1A, and H2, along with U2E. For genetic context, these haplogroups are overwhelmingly concentrated today in the Caucasus region, the mountainous territory between the Black Sea and the Caspian Sea, encompassing modern Georgia, Armenia, and Azerbaijan. One particularly striking result came from the most elongated skull in the sample set, whose bone powder came back as haplogroup T2b, a lineage that originates in Mesopotamia in what is now Syria and Iraq, the heart of the ancient Fertile Crescent. The blood types of the sampled Paracas individuals were also anomalous. Native American populations are expected to be overwhelmingly type O. The Paracas samples showed high percentages of type A, some type B, and significant type AB. These are blood types associated with ancient Eurasian populations, not with the indigenous Americas. Some of the skulls still have hair attached. That hair is red or blonde, colors that simply do not occur naturally in indigenous South American populations. 2 independent hair analysis experts have confirmed that the coloration is not a product of age, decomposition, or environmental bleaching. That hair was red when the person was alive. Now it is critical to be honest about what science does and does not know here. These DNA results, while genuinely surprising, were not published in peer-reviewed journals with the full rigor of modern genomic sequencing. The chain of custody for the samples has been questioned. Ancient DNA degrades, contaminates, and fragments in ways that can produce misleading results, particularly when specimens are handled outside controlled laboratory conditions. Mainstream archaeology treats the Paracas' DNA claims with deep caution, and that caution is scientifically justified. What the results suggest, the presence of Caucasian and Middle Eastern lineages in pre-Columbian Peru, would require extraordinary evidence to confirm, but it would also require a mechanism, and that mechanism might be stranger than anyone expected. For decades, the dominant theory of how humans reached South America was simple. They crossed Beringia, the land bridge connecting Siberia to Alaska, sometime between 15 and 20,000 years ago, then gradually moved south along an ice-free corridor through what is now Western Canada. This Clovis First model, named after the distinctive stone tools associated with early North American inhabitants, was the unchallenged orthodoxy of American archaeology for most of the 20th century. Then the genetic and archaeological evidence started falling apart in spectacular fashion. Archaeological sites in South America, Monteverde in Chile, Pedra Ferrada in Brazil, were dated to periods that would have been impossible if humans entered North America only 14 or 15,000 years ago. The math simply didn't work. People can't walk from Alaska to southern Chile in a few centuries, while also establishing complex campsites, hunting megafauna, and developing regional cultures. Something was wrong with the timeline. Modern ancient DNA research has now produced a different picture. Evidence increasingly supports what is called the Beringian standstill hypothesis, the idea that the ancestors of the first Americans didn't march straight through Beringia on their way south. Instead, they stopped for a very long time. Genetic analysis suggests that a founding population was effectively isolated in the Beringia region for somewhere between 5,000 and 15,000 years, accumulating unique genetic mutations that are found nowhere in Asia and only in the Americas. This period of isolation, during the height of the last glacial maximum approximately 21,000 years ago, created a genetically distinct people. Then, as the ice retreated around 17,000 years ago and the Pacific coastline became accessible, these people moved, not through an inland corridor, down the coast, by boat and on foot, hugging shorelines that offered food, water, and navigation. The coastal route hypothesis has been transforming our understanding of the Americas for years. It explains how humans reached South America so early. It explains the genetic diversity found in ancient South American remains, and it connects to a growing body of evidence that ancient populations were far more mobile, far more maritime, and far more globally connected than the old Beringia to Kansas textbook story ever suggested. And what does all of this have to do with a mummy? The word mummy conjures images of Egypt, of linen wrappings and golden masks, but Peru has been producing mummies for far longer than Egypt, in far greater numbers, and under conditions that preserve biological material in ways that continue to astonish researchers. The cold, dry air of the Andean highlands and the extreme aridity of the Pacific coastal deserts create natural mummification conditions without any chemical intervention. Bodies buried in these environments simply dry out and endure. Peru has yielded mummies dating back over 9,000 years. The oldest known mummies in the world, predating the earliest Egyptian examples by more than a millennium, were found along the northern coast of Chile and Peru, created by the Chinchorro culture using techniques of extraordinary sophistication. These weren't accidents of climate. These were deliberate, repeated rituals of preservation carried out by a people who understood, at some profound level, that the dead had things to tell the living. They were right. They just had to wait for the technology. What modern ancient DNA sequencing has done to Peruvian mummies is nothing short of revolutionary. Every tooth, every hair root, every fragment of preserved bone is now a potential data point in a global genetic map that is being redrawn in real time. The Chincha Valley study used genome-wide sequencing, analyzing hundreds of thousands of individual genetic markers simultaneously across 21 ancient individuals. That scale of analysis would have been inconceivable 20 years ago. Today it runs on machines in university laboratories, processes data through bioinformatics algorithms, and produces family trees, migration maps, and ancestry breakdowns with a level of precision that makes old archaeological guesswork look almost quaint. And the more data that comes in from Peru, the more complex and surprising the picture becomes. A 2024 study published in Scientific Reports sequenced the complete mitogenome, the entire mitochondrial genetic code, of a 500-year-old Inca child mummy and found that it belonged to a haplogroup called C1bi, a rare genetic variant that may have been far more widespread in ancient Peru than in any surviving modern population. The variant traces its origins to approximately 14,000 years ago and suggests ancient Peruvian populations harbored genetic diversity that has since largely vanished from the living human gene pool. These are ghost lineages, ancestral genetic lines that flourished for thousands of years and then disappeared, surviving only in the teeth and bones of the preserved dead. A separate study tracked the genetics of Peruvian highlanders going back 6,000 years and found evidence of complex population shifts, bottlenecks, and replacements that don't correspond to anything in the archaeological record. People who should have been there weren't. People who shouldn't have been there were. The mountains of Peru, it turns out, witnessed waves of genetic replacement and migrations that archaeology had no way of detecting, because it leaves no stone tools, no pottery, no visible signature in the sediment. Only the DNA remembers. This is what makes Peru so uniquely important in the story of human history. It sits at the end of the longest land migration in the entire history of Homo sapiens. Every person who ever walked the full length of the Americas from Beringia to the southern tip left traces of that journey in the genetics of the people who settled along the way. Peru is not just a destination, it is an archive. Every mummy is a chapter. Every tooth is a word in a sentence that is finally, after thousands of years of silence, being read aloud. And what that sentence is saying will take generations of researchers to fully translate. But some things are already undeniable. The Americas were not populated in a single wave of migration by a single genetically homogeneous group of Siberian hunters. Multiple waves from multiple source populations arrived at different times through different routes and mixed, separated, remerged, and diversified in ways that produced the extraordinary genetic and cultural complexity of pre-Columbian civilization. Peru's coastal communities were not isolated, parochial cultures locked to their immediate geography. They were mobile, ambitious, long-distance travelers who moved hundreds of kilometers, maintained their identities across generations, intermarried strategically, traded broadly, and built social networks of a scale and sophistication that no one suspected until a genome sequencer proved it. The old story said, people came, people stayed, empires rose. The DNA says something else entirely. It says, people came, people moved, people mixed, people remembered. People crossed deserts and climbed mountains and paddled coastlines for reasons that were deeply human. Survival, opportunity, family, identity. And did all of this not once, but repeatedly over thousands of years, leaving traces of every journey written into the bones of their descendants. One of those descendants is now lying in a laboratory somewhere, preserved by desert air for 8 centuries, still carrying red pigment on her cheek, still holding the spinning tools of her northern homeland, still inscribed with the evidence of a child she brought into the world on a journey that history forgot, science remembered. And that is the most extraordinary thing about what ancient DNA is doing to the history of the Americas. It is not just rewriting the textbooks, it is restoring the people. Not as anonymous prehistoric populations defined by the stone tools they dropped or the pots they fired, but as individuals, families, communities, people with identities they fought to preserve across centuries and continents, people whose specific choices and specific lives shaped the world we live in today, people who left no written record but whose bodies, against all probability, survived long enough for us to finally learn their names. The DNA from a Peruvian mummy just rewrote the history of the Americas. And it's only just getting started. But before we can truly appreciate the scale of what is being rewritten, we need to talk about something that most mainstream science channels will never tell you, because it sits right on the uncomfortable boundary between established science and the kind of finding that makes academic institutions deeply, visibly nervous. Go back to the Spirit Cave mummy. In 1940, in the Nevada desert, archaeologists opened a carefully wrapped bundle and found inside a human body that had been naturally mummified for approximately 10,000 years, making it older than Ötzi the Iceman, Europe's famous alpine mummy, by roughly 3,000 years. The Spirit Cave mummy was quietly catalogued, stored, and largely forgotten. For decades, the prevailing scientific opinion was that ancient remains like this one were not related to modern Native Americans. The dominant theory held that the earliest inhabitants of the Americas, the so-called Paleo-Americans, were a genetically distinct population, separate from and replaced by the ancestors of today's indigenous peoples. This was not fringe thinking. This was taught in universities. It was the official position of major archaeological institutions. The Falan Paiute Shoshone tribe, whose ancestral territory includes Spirit Cave, repeatedly and passionately contested this view. They argued that the mummy was their ancestor, that they had spiritual and cultural obligations to him, and that he deserved to be repatriated and given a proper burial according to their traditions. The Bureau of Land Management refused for 2 decades, clinging to the scientific establishment's position that the biological link between Paleo-Americans and modern Native Americans was unproven. The legal battle stretched on through the 1990s and into the 2000s, a painful and humiliating standoff between a tribe fighting for their ancestor and institutions more committed to protecting their theories than listening to the people who had always known the truth. In 2016, a team of geneticists finally analyzed the Spirit Cave mummy's DNA. The results were unambiguous. The individual was directly and undeniably genetically linked to modern Native Americans, not distantly related, not a precursor population that branched off before the main migration, a direct ancestor. The Bureau of Land Management changed its position almost immediately. In 2018, the Spirit Cave mummy was repatriated to the Falan Paiute Shoshone tribe. What this episode reveals is something that goes beyond genetics. It reveals the extraordinary human cost of getting the history of the Americas wrong. For 2 decades, a community of living people was denied the right to bury their dead because the scientific establishment had built a model that couldn't accommodate indigenous oral tradition, couldn't accommodate the spiritual claims of people who had lived on this land for 10,000 years, couldn't accommodate the possibility that the people most likely to know the truth about ancient American history were the people whose grandparents' were that ancient history. Ancient DNA is correcting all of that, not just by revising academic papers, by physically returning ancestors to the communities who never stopped claiming them. Now think about what that means for Peru. The Inca Empire, which the Spanish dismantled with terrifying efficiency beginning in 1532, was the culmination of thousands of years of civilization building along the Pacific coast and in the Andean highlands. But underneath the Inca were the Wari, and underneath the Wari were the Tiwanaku, and underneath them were the Nazca and the Paracas, and the Moche and the Chavin, cultures of extraordinary sophistication whose origins, relationships, and genetic connections have remained largely invisible to conventional archaeology. Ancient DNA is now illuminating all of them simultaneously. A study of highland Peruvian populations dating back 6,000 years found that the genetic makeup of highland communities shifted dramatically at certain points in time. Not gradually, as you'd expect from slow cultural diffusion, but abruptly, suggesting actual population replacement. People who had lived in the highlands for generations were genetically displaced, replaced by communities moving up from the coast or down from even higher altitudes for reasons that remain unclear, but are almost certainly tied to the same forces that drove the Chincha Valley migration: climate events, political pressure, resource access. The Wari Empire, which dominated the Peruvian highlands between roughly 600 and 1000 AD, shows up in the ancient DNA record as a period of significant genetic mixing across previously isolated populations. This was not just cultural conquest, this was biological convergence, as the expansion of Wari influence brought genetically distinct communities into sustained contact for the first time. Haplogroup analysis of Wari period individuals reveals ancestry connections, stretching from the northern coast to the southern highlands to the Amazon basin, genetic threads tying together populations that had no reason to be connected based on what archaeology alone could tell us. And then there is the DNA of disease. One of the most devastating things ancient DNA has revealed about pre-Columbian Peru is the presence of pathogens that modern medicine barely recognizes as ancient. A 500-year-old Inca mummy, initially assumed to be a German bog body before stable isotope analysis placed her firmly on the Peruvian coast, was found to have suffered from an advanced chronic trypanosoma cruzi infection, the parasite responsible for Chagas disease, a condition that attacks the heart and digestive system over years or decades, eventually causing catastrophic organ failure. The evidence was preserved in her intestinal tissue, her massively hypertrophied heart muscle, and the microscopic parasites still visible under histochemical analysis. She also showed signs of catastrophic blunt force trauma to the face, applied with such force that it destroyed the bones of her forehead and cheeks without any evidence of healing, meaning it killed her. The researchers who analyzed her remains proposed ritual homicide, a sacrifice, perhaps connected to the severity of her illness, perhaps for other reasons that the bones cannot fully explain. She was not unique. Human sacrifice in the Inca world is well documented archaeologically, and ancient DNA is now revealing details about these individuals, their diet, their migration history, their health, their genetic background, that transform them from anonymous ritual victims into specific human beings with stories that deserve to be told. The Capacocha ritual, in which children were sacrificed at high-altitude mountain shrines, produced some of the best preserved mummies ever found, including 3 child mummies discovered on the summit of Argentina's Lulailico volcano, at an altitude of over 6,000 meters. DNA and isotope analysis of these children showed that they had consumed increasing amounts of coca leaves and alcohol in the months before their deaths, a ritualized preparation documented in historical sources. One of the children had a parasitic lice infestation. Another showed signs of a pulmonary infection. They were sick, sedated, and carried to the top of the world to die at the command of an empire. And their bodies preserved every detail of that final journey in biological code that survived 5 centuries of freezing altitude. What do you do with that knowledge? What does it mean to recover the biological reality of a child who died in a religious ceremony 500 years ago, whose body was considered sacred, whose death was considered a gift, and whose DNA now tells us she was also just a sick little girl who had caught a cold? This is the ethical frontier that ancient DNA is forcing researchers and communities alike to confront. The science doesn't care about categories. It doesn't distinguish between sacred remains and archaeological samples. It reads the molecule and reports what it finds. Peruvian law now requires that any exportation of ancient DNA samples, any fieldwork, and any laboratory analysis be conducted under permits issued by the Ministry of Culture, in collaboration with descendant communities and governing agencies. The Chincha Valley study was conducted under exactly these conditions. But the question of who owns ancient Peruvian DNA, who has the right to sequence it, publish it, and profit from the knowledge it generates, is one that science is still fumbling its way toward answering. And the history of the Spirit Cave mummy is a reminder of exactly what is at stake when institutions get that question wrong. Here's the final thing that the DNA from Peruvian mummies is teaching us, and it might be the most important lesson of all. We are not the first people to grapple with complexity. We are not the first people to navigate displacement, migration, cultural loss, and cultural preservation simultaneously. The people whose bones are being sequenced in laboratories today faced versions of every challenge we face now. Climate instability, political upheaval, the collapse of familiar systems, the need to build new communities in unfamiliar places while holding on to the things that made them who they were. They managed. A woman walked 700 kilometers from her northern homeland, carried her culture and the pigment she wore on her face and the tools she spun fiber with, gave birth, raised a child, and was eventually buried in a valley far from where she was born. Her identity intact, her story preserved in the microscopic language of her own genetic code. We found her, and she has things to tell us. The history of the Americas is not a story that has been told. It is a story that is being discovered. Every mummy, every tooth, every strand of ancient hair is a voice speaking out of a silence that lasted for centuries. Genomic science has given us the ability to finally listen. And what we are hearing, from the Chincha Valley to the Paracas Peninsula, from the peaks of the Andes to the deserts of Nevada, is a story of human movement, human resilience, and human complexity so vast and so rich that the old textbook version of it looks, in comparison, like a child's drawing of the ocean. The DNA from a Peruvian mummy just rewrote the history of the Americas. Expect the next chapter very soon.